Cyclic AMP and calcium ions Jillian Radford, Jodianne Brown, Soraya Rajendran. Second messengers are small, non-protein, water-soluble molecules or ions that spread throughout a cell by diffusion. Internal signaling molecules released due to external first signals. Trigger sub-response pathways. Cyclic AMP and calcium ions are common second messengers, and cyclic AMP CAMP is one of the most widely used second messengers. Cyclic AMP CAMP. A typical second messenger that affects metabolism. Many signal molecules trigger formation of CAMP. CAMP is a second messenger synthesized by K protein. Cyclic adenosine monophosphate, cyclic AMP or CAMP, is a small molecule made from ATP, and they relay as well as amplify the intracellular signal in response to signals. An enzyme called adenylyl cyclase converts ATP into CAMP, removing two phosphates and linking the remaining phosphate to the sugar in a ring shape once generated. CAMP can activate an enzyme called protein kinase A, pKa, enabling it to phosphorylate its targets and pass along the signal. Protein kinase A is found in a variety of types of cells, and it has different target proteins in each. This allows the same CAMP second messenger to produce different responses in different contexts. CAMP signaling is turned off by enzymes called phosphodiesterases, which break the ring of CAMP and turn it into adenosine monophosphate, AMP. Adenylyl cyclase, an enzyme in the plasma membrane, rapidly converts ATP to CAMP in response to a number of extracellular signals. The immediate effect of CAMP is usually the activation of protein kinase A, which then phosphorylates a variety of other proteins. Cyclic AMP is involved in the regulation of glycogen, sugar, and lipid metabolism, various cellular functions, including cell growth and differentiation. Gene transcription and protein expression. Increased CAMP, through its coupling with other intercellular messengers, increases contractility, inotropy, heart rate, chronotropy, and conduction velocity, dromotropy. In some cases, increase in levels of CAMP may result in an increase in the production of a neurotransmitter, contributing to an agonist effect. If CAMP-dependent pathway is not controlled, it can ultimately lead to hyperproliferation, which may contribute to the development and/or progression of cancer. CAMP is a key second messenger that regulates function of proteins involved in ion homeostasis and cardiac excitation contraction. Coupling. Cyclic AMP second messenger mechanism of water-soluble hormones. Step 1. The hormone or first messenger binds to receptor. Step 2. Receptor activates G protein. Step 3. The G protein activates adenylate cyclase. Step 4. Adenylate cyclase converts ATP to CAMP, second messenger. Step 5. CAMP activates protein kinase. Cyclic AMP CAMP is a protein messenger present in many different types of cells, which activates CAMP-dependent protein kinases, causing them to transfer phosphate groups from free molecules of ATP to various proteins in the cell. In this case, protein kinase A, pKa, transfers these phosphate groups to proteins on ion channels on the cell membrane. Phosphorylating these channel proteins increases the chances of the channels opening, thus increasing the chances of depolarizing the postsynaptic neuron at the axon hillock and setting off an action potential. Such events are called excitatory postsynaptic potentials, EPSPs, in this case when dopamine receptors are activated by dopamine. The linked G protein dissociates and stimulates the production of CAMP, which then activates the protein kinase, which in turn phosphorylates the ion channel, allowing sodium Na+ ions to rush into the postsynaptic call and depolarize it. If enough depolarization occurs, An action potential will be generated and a signal sent down the axon of the postsynaptic cell. G proteins, also known as guanine nucleotide binding proteins, are a family of proteins that act as molecular switches inside cells and are involved in transmitting signals from a variety of stimuli outside a cell to its interior. CAMP is a common second messenger that is regulated by the activation of G protein-coupled receptors, GPCRs, and mediates numerous biological responses. Activated GPCRs cause a conformational change in the attached G protein complex, which results in the G's alpha subunits exchanging GDP for GTP and separation from the beta and gamma subunits. The G's alpha subunit, in turn, activates adenylyl cyclase, which quickly converts ATP into CAMP. The alpha subunit of the G protein is activated by replacing GDP with GTP. When the activated alpha subunit binds to the calcium ion channel, the channel opens in. Calcium ions enter the cell. In most cells, the concentration of calcium ions, Ca2+, in the cytosol is very low as ion pumps in the plasma membrane continually work to remove it for signaling purposes. Ca2+ may be stored in compartments such as the endoplasmic reticulum in pathways that use calcium ions as a second messenger. Upstream signaling events release a ligand that binds to and opens ligand-gated calcium ion channels. These channels open and allow the higher levels of Ca2+ that are present outside the cell or in intracellular storage compartments to flow into the cytoplasm, raising the concentration of cytoplasmic Ca2+. Calcium ions, Ca2+, act as a second messenger in many pathways and can act directly from the activation of ion channels causing signal transduction. Calcium is an important second messenger because cells can regulate its concentration. Calcium can act directly from the activation of ion channels causing signal transduction. It can also act as a second messenger from signal transduction pathways coming from external receptors such as G protein-coupled receptors. A signal relayed by a signal transduction pathway may trigger an increase in calcium in the cytosol pathways, leading to the release of calcium-involvinocetyl triphosphate (IP3) and diacylglycerol (DAG) as additional second messengers. They play an important role in signal transduction pathways where they act as a second messenger in neurotransmitter release from neurons in contraction of all muscle cell types. And in fertilization, some proteins in the cell have binding sites for Ca2+ ions, and the released ions attach to these proteins and change their shape and thus their activity. The proteins present and the response produced are different in different types of cells. For instance, Ca2+ signaling in the beta cells of the pancreas leads to the release of insulin, while Ca2+ signaling in muscle cells leads to muscle contraction. Ca2+ is important for cellular signaling. For once it enters the cytosol of the cytoplasm, it exerts allosteric regulatory effects on many enzymes and proteins.